This video will discuss a experiment we just finished several weeks ago, which is an experiment about knock out a gene from a fungus. This fungus is called Aspergillus nigerli. Firstly is a knockout process, which is the knockout process defects a gene function in a species, and the cassette is a sequence. In this vector, the cassette sequence on the two ends, which is able to bound to specific site on the strand that we are interested in. The cassette sequence will directly bound to the site on the Aspergillus nigerli. This is the gene that we are interested and want to knock out in the strand. And in this sequence, this is the cassette sequence that will bound and identify the sequence of the gene. And the cassette sequence is widely used to replace the gene. For example, the cassette gene usually able to bound to the vector, and the markers and our interest gene will involve. There may be a promoter on the upper strand of our interest gene. But in this situation, we do not want to introduce or replace the gene. We want to knock out the gene between the two cassettes. That means in this sequence, there is a gene between the cassette sequence. If we replace this sequence with our vector, that will definitely defect the function of the gene here. So in this experiment, we do not need a gene region. We just need a marker and two cassettes on the end. It will replace some sequence of the gene and defect the gene. As a result of the effect of the gene being knocked out. In this vector, the marker is called AF-PYRG+, and this gene encodes an enzyme called adenine 5' phosphate carboxylase, which is an enzyme that involves in a cascade that produces uridine and uracil. The marker is called AF-PYRG+, and this marker is a DNA code was isolated from the another species which is called Aspergillus fumigatus, which is in the same phylum but it's a different species. Looks and its appearance looks different from Nigerlands. In this experiment, the strand Aspergillus nigerli was a strand that do not have the also adenine 5' phosphate carboxylase related genes. So the strand is unable to survive in the media without uridine and uracil. So if the cassette being successfully inserted, the strand will survive in the media without uridine and uracil. So in this media, we are able to select the successfully knockout strand. After the cassette vector knock out the genes, we inoculate on another plate and form a strand with pure gene. So with a strand with the same genetic information. After that, we want to further prove the genes being introduced are being knocked out. We want to make sure the genes being knocked out, so we use PCR to amplify the genes with a forward primer on the prime cassette, and the reverse primer. We have two reverse primer. One is a primer at AFPYRG plus side, another reverse primer on the original gene side. If the genes been knocked out, there will be no reverse primer. The reverse primer will not bind to the gene, do not have any target to bind. If the gene is not been knocked out, the reverse primer. If the genes been knocked out, there will be no DNA being amplified with the original gene reverse primer. If the gene didn't be knocked out, there will be some positive result there will be some positive result on gene reverse primer, original gene reverse primer. In this gel, first one is the G2, the group 2's result. In this experiment, in the left side, from the left side to the right side, the first column is a marker that induces the molecular weight. First line is 100 base pair and 100 base pair and 200 base pair, 300, 4, 5, 6, 7, 8, 9, 10. But there's some mistake in the there are some unexpected result here. There are two lines because there's some situations there. Because in the process of running the gel, they cut off the electric and run the gel. So there may be some unexpected result there. Maybe need for further study. But in theory, the target being the DNA being amplified should be there in around 1000 base pairs. And every group we have three sets. One is a gene with a primer on the marker and with a reverse primer on the marker. Another set is with three sets. One set has a reverse primer on the marker. Another one have the reverse primer on the original gene DNA. So, and there are three sets of them. Every group have three sets. In our group, they are all negative, and we left the control group. We only have the control group, so there's some big mistake here. In group 12, two of them succeed, and the left one didn't work, so there are some failure rate. Okay, thanks for watching. That's all for the experiment.